Hi, welcome to Thinkpharma YouTube channel. After our first class, so this is our second class. So moving into the topic that is alkaloids. So coming to the definition of alkaloids, what are alkaloids? So these alkaloids are organic products of natural or synthetic origin which are basic in nature. So what is meant by basic? So a compound is basic, acidic or neutral. It will be in three forms, right? So these alkaloids are basic in nature, and how we can say these are basic? So in these nucleus, in the structure of that alkaloids, those are having nitrogen atoms in their ring system. Okay, so these are basic in nature with one or more nitrogen atoms of a heterocyclic nature, and these possess specific physiological actions on human or animal body. So when it was used in small quantities, it used to show its activity. So heterocyclic nature, nothing but, so if a benzene is there, in that benzene all things will be carbon only, right? So heterocyclic means other than C, like N, S, O, things are there. So those are heterocyclic in nature. Okay. If we will attach N or S or O, so to the benzene ring, so that it will become the heterocyclic nature. Okay. So clear, right, with the definition. So moving on to the different types of alkaloids. There are three types of alkaloids, and then first one is true alkaloids. So these two alkaloids which were derived from amino acids, and they contain nitrogen atoms in their heterocyclic ring. Okay, already we have discussed in the definition, right? And the ring system which is containing n atoms, so those are called as heterocyclic ring. So examples are atropine and morphine. And coming to the proto-alkaloids here, these are derived from the amino acids, and it do not contain nitrogen atom in the heterocyclic ring. Okay, so coming to the examples, these are ephedrine and mescaline. And then pseudo-alkaloids, these are not derived from amino acids, but it contains nitrogen atom in their heterocyclic ring. Okay, and then example is caffeine and capsaicin. So moving on to the properties of these alkaloids. So these alkaloids are solid crystalline substance. Okay, even we can find some liquid alkaloids. Those are nicotine and quinine. Okay, and then these alkaloids are colorless. Even some alkaloids we can find which are colored. So for example, berberine, which was shown in yellow color, and salts of sanguinolene, which were copper red color. So these alkaloids are having sharp melting point, and these alkaloids are soluble in organic solvents, but these are insoluble in water. Okay, and these alkaloid salts are soluble in water and insoluble in alkaloid organic solvents, and these alkaloids are getting degraded easily. Okay, if we will expose these alkaloids to light and air, so these used to get degrade fastly. And then these alkaloids are basic in nature, as we already know. These are basic in nature due to the presence of non-pair of uh, electron on nitrogen. And then these alkaloids are neutral acidic when adjacent functional groups are electron withdrawing. Already we have learned that these alkaloids are basic in nature, right? When these alkaloids can be neutral and acidic also when it was functional groups are electron withdrawing. Okay, and this example is amide group. And so moving on to the decomposition of alkaloids, as in properties we have already discussed, these alkaloids can easily degraded when we expose it to air and light. So how these cause degradation means, so these are basic in nature, right? So these are very sensitive to decomposition, and it causes a problem during the storage. Because of this reason, they are made into salt form and stored. So if these alkaloids are in salt form, no, it will not get degraded. If other than in salt form, the alkaloids will get degraded when exposed to light and air. Okay, and then um, the alkaloids are distributed in various parts of plant, like as we have already discussed in introduction. the phytopharmaceuticals or chemical constituent which were derived from different sources of natural things like plants, animals, minerals, all that we have learned. And these alkaloids are distributed in several parts like leaves, flowers, bark, roots, rhizomes, seeds, all these things. So for example, in leaves, belladonna, datura, tobacco, vinca, and coca, all these things were found in leaves. And cinchona and kudsi are the alkaloids where we can find it in bark. And then Rauwolfia, ipecac, and aconite are the things where we can find in the roots and rhizomes of the plant. And then black pepper, as we all know, those are fruits. And opium, which are the alkaloids were present in fruits. And then nux vomica and colchicum are the seeds. And then whole aerial part, ephedra and lobelia, etc., are the things, alkaloids, which were distributed in several parts. And then how we will extract these alkaloids from different parts of the plant. So coming to the method A, this is a stas auto method, is the main important method for extraction of these alkaloids. So this method is based on distribution of alkaloidal bases between acid or aqueous solution and immiscible organic solvent. Okay, and then in the first step, powder material containing alkaloidal salt are defatted with non-polar solvents and moist with water and treated with ammonia. So first we have to take this powder material where alkaloids are present, and then it was defatted with non-polar solvents. Like non-polar solvents are like hexane, petroleum ether. These are non-polar solvents. So if when we will use non-polar solvents to this powder material, all the fatty compounds will be extracted here in these non-polar solvents and moist with water, and then treat with ammonia. And then in next step, then extract is obtained by mixing of organic solvent. So after defatting with this non-polar solvent, so the extract is mixed with organic solvent like chloroform ether, and we have to concentrate it, means we have to evaporate the solvent by using some rota evaporator or something. Okay, then dissolve the alkaloid salt and basify with ammonia. So after mixing the extract with organic solvent and after concentrating, the next step is the alkaloid content or the concentrated extract is there, no, that thing we have to make basify with using ammonia. Okay, and then final step is finally organic phase free alkaloids were obtained, and then dry, we have to dry them. Okay, and then moving on to the method B, this is second method of extraction of alkaloids. So powder material was extracted with water and aqueous alcohol containing dilute acid. So in the first method A, we are taking non-polar solvent. So like hexane and petroleum ether, those things. And here in method B, we are taking water and aqueous alcohol containing dilute acid. We are taking polar substance. Okay, and then after this step, shake with chloroform and other organic solvent, and here we will get two products like chloroform layer and then aqueous layer. So here we are using chloroform and organic layer, right? So when we will mix these two layers, those are immiscible, so we have to separate it. So chloroform layer will be separated with pigment and other unwanted material. So all the fatty things, all the uh, unwanted things whichever will be there, that will be gone through this chloroform layer. And this aqueous layer, it we have to add sodium bicarbonate or ammonia to this aqueous layer, so then free alkaloid will get precipitated, and then we have to concentrate, and finally we can collect our alkaline salts, alkaloid content. So here coming to the classification of heterocyclic alkaloids, we already know, right, types of alkaloids like true alkaloids, pseudo alkaloids, proto alkaloids, as we have discussed before slides. So this is the classification of heterocyclic alkaloids, that is true alkaloids based on their chemical nature. So there are different kinds of alkaloids like pyrrole alkaloids, piperidine alkaloids, imidazole alkaloids, quinoline alkaloids, pyrrolidine alkaloids, pyridine alkaloids, isoquinoline alkaloids, indole alkaloids, purine alkaloids, tropane alkaloids, quinazoline alkaloids, norlupinane alkaloids. So these are the examples and even biological source and the family uses. These are very much important for these competitive exams. So in competitive exams they will ask like pyrrole alkaloid is an example of which family, or pyrrole alkaloid is an what is the biological source of this high green, like that they will ask in these competitive exams. So we should show more interest and importance in studying all these types of alkaloids and these biological sources, examples and family. All these things were very much important. And then moving on to the other slide, this is classification of non-heterocyclic alkaloids, pseudo and proto alkaloids. So before we have done classification of heterocyclic alkaloids, that is true alkaloids, right? And this is classification of non-heterocyclic alkaloids, that is pseudo and proto alkaloids. So coming to these examples are phenylethylamine and steroidal alkaloids, tropolone alkaloids. All these are the examples of pseudo and proto alkaloids. So as we have discussed before, you have to be ready and you have to give more importance to type of alkaloids, biological sources and its family. And then moving on to the next slides. So based upon their chemical structure, these alkaloids are of two types. One is atypical alkaloids, that is these are known as non-heterocyclic alkaloids and contain N in aliphatic. N means nitrogenous compounds in the aliphatic chain. Okay, these are atypical alkaloids. And second thing is typical alkaloids. So these are also known as heterocyclic alkaloids and contain N in heterocyclic ring. So first one is atypical alkaloids, where non-heterocyclic alkaloids and contain N in aliphatic chain. Okay, in the ring system N will not be there. Only in aliphatic system N will be there in atypical alkaloids. But in typical alkaloids, heterocyclic alkaloids, and that heterocyclic ring contains N in the structure. So how we will identify this alkaloids? So there will be many tests, right? So to identify proteins, to identify some glycosides, to identify some alkaloids, there are specific tests which we have to perform to confirm this is an alkaloid. So coming to the first test, that is Mayer's test, where this alkaloid drug is treated with potassium mercuric iodide. It gives cream white color precipitate. And then next is Dragendorf test, where our alkaloid drug is treated with potassium bismuth iodide. It will give orange red color. Okay, and then Wagner's test, our alkaloid drug is treated with dilute iodine solution. It gives reddish brown precipitate. And then Hager's test, our alkaloid drug is treated with picric acid, then it will give crystalline yellow color precipitate. And then finally tannic acid test, where this alkaloid drug is treated with few drops of tannic acid solution, so it will give the buff color precipitate. So in competitive exams you will get these tests, like Mayer test is responsible for which color, and what is the chemical that is present in Mayer's test, or what is the chemical that is present in Dragendorf test, and if alkaloid drug is treated with potassium bismuth iodide, which color it will give. Like that we will get questions from these identification tests. And so all these identification tests were very important to secure good marks. And then some more important, particular important color reactions. So vitally Morin test, it will give positive test for Solanaceae family, such as drugs like belladonna, datura, henbane, tobacco, etc. So our alkaloid is mixed with fumic nitric acid, and then it should be evaporated to dryness, and then after drying it was dissolved in acetone and added a methanolic solution of KOH. Then it will give violet color. So it will be representing tropane group was presenting this compound which they have given. So it is a confirmation test. And then next is Van Oork's test. This is important and it will show positive for ergot alkaloids. So sample react with para-dimethyl amino benzaldehyde and it will and dilute sulfuric acid. Then it will give blue color. So our alkaloid drug is treated with para-dimethyl amino benzaldehyde and dilute sulfuric acid. Then it will give blue color. So this is indicating indole group was present. And then one more test is Freud's test. So it is giving positive test for opioid alkaloids. So our sample reacts with Freud's reagent, that is sodium molybdate in concentrated sulfuric acid, and it forms brownish black color, and it represents isoquinoline group is present in this compound like that. And then thalloquine test, it is giving positive to quinine alkaloid. And then the uses of alkaloids. So these alkaloids are having many physiological activities that we have discussed in the first slide. So these alkaloids are having many physiological activities and it was showing many useful uh, things to treat many several other disorders and diseases. So example like analgesic example is cocaine, morphine, codeine are the things which are used as analgesic, and reserpine, deserpine are the things which are used as anti-hypertensive, and quinine, cinchonine are the drugs which are used as anti-malarial, and vinblastine is the drug which was used as anti-tumor, and codeine and noscapine are the alkaloids which were used as anti-tussive, and quinidine is an alkaloid which was used as cardiac depressant, and ergometrine and ergotamine are the alkaloids which were used as oxytocic, and theobromine is an alkaloid which is used as a diuretic, and theophylline is an alkaloid which was used as smooth muscle relaxant. So that's all for today's alkaloids class, and the next will be continued in next video. So until then, keep watching our Think Pharma YouTube channel. Please like, share, subscribe. For more details, follow Think Pharma YouTube channel. And finally, thank you.